Yes or no? It says
Recording?
recording.
Yeah, we're recording.
Okay.
So where were we?
Well, this is August 2nd, 2021. Let's just, start that way. Where were we? Well, I think we were talking about rhythmicity and recruitment and that kind of thing, or was that an earlier?
Well, that was related to it. So
And then focus and
well, I guess what we were like exploring was the different roles that uh, different kinds of like rhythmic or like more or less rhythmic or arrhythmic or more or less ordered or disordered processes could have like in biology, and then trying to work our way around to understanding how these things relate to like affect, pleasure, pain, and consciousness. And so like trying to go from like what are the principles of dynamic adaptive order in biology, and what can they tell us about the nature of experience? It seems like the general shapes was a big space we were exploring.
yeah. Well, I think, I mean, what when you talk about order, and it's the idea of an optimal level, because if it gets too ordered, then that's a seizure. When there's massive synchronous firing of neurons, then that's a seizure, and that is maladaptive, because what seems to me like what is required in the idea of ordering is an optimal degree of ordering and diversity. I mean, a diversity of like, being in yourself, being aware of yourself, but also being aware of the environment. And so that is an optimal level of functioning, as opposed to hypersynchrony, which would be a seizure in which everything is you need a certain degree of diversification of awareness, an optimal diversification of awareness. But in a seizure, it's all something, and that's suboptimal, because it's and I keep thinking, I think one of the most important concepts for me is the work that Donald Lindsley did, where he explained the change in EEG pattern from sleep to arousal. And what he was saying, and it was, I think, because what he explained it in terms of activity in subpopulations of neurons. And if there's like one big population of neurons all doing the same thing, that is a seizure. And in sleep, there are there are relatively few and large subpopulations of neurons. Geographic, he has like a geographical map on the brain, on the cortex, where so there are there are relatively few subdivisions, and they're firing at different phases. So that generates a high amplitude slow wave. When you do the algebraic sum of those several relatively few subpopulations, there and you algebraically sum the depolarizations, say, you get large amplitude because it's large populations firing together, and relatively slow because when you add all the phases together, there are relatively few phases. So the whole thing, you have large amplitude because of synchrony and relatively slow waves because you have several subpopulations at different phases. But then during arousal, what happens, as in his idea, is that those relatively few, so you get a delta wave, say, in sleep, because it's high amplitude, slow waves. When you, but then during arousal, what happens is that these relatively few subpopulations break up into many more subpopulations, all going at their different phases. So when you add those algebraically, you have relatively low amplitude because they're all in different phases and lots of different subdivisions. And when you add their phases algebraically over time, it looks like the frequency has increased because they're all out of phase with each other. But the point of that is that because each subunit has an optimal moment of processing, of sensitivity to stimuli or to responses, that means that you have many more options for response and for awareness and response, motor response, when aroused, compared to during sleep or seizure. So the change in EEG from high amplitude slow waves, of course, with a seizure, it's ultra high amplitude, ultra slow, spikes and domes. That's the extreme. But then during sleep, it's the high amplitude slow waves. And during arousal, it's the low amplitude fast waves, because the whole idea is that a population of neurons is optimally sensitive to input and to output at particular phases of that activity pattern. So I think that really explains the change in brain activity. From showing that is an optimal thing with the concept of a optimal sensory response and optimal motor response at particular phases of an EEG pattern, of an EEG activity pattern. So that's what the rhythmicity is, that the different subpopulations have their rhythmicity, like in the alpha rhythm, or in gamma, where gamma is even beta and gamma, it's even higher, it's even more distributed,
more diverse,
right? So arousal is really a diversity,
but not completely without order. It's just there's a greater range of options, like each one might have its time in a particular context. And this
and he showed, and there are lots of studies, and I reviewed I published a whole paper on rhythmicity. There are lots of studies showing, very, very elegant studies, and he himself showed that when he measured the alpha rhythm, and he asked people to press a button when he flashed the light, he flashed a strobe light at various phases of the alpha rhythm. And he asked the people to press the button as fast as they could when they saw the flash of light. And it was just a strobe flash at various times, seconds in between them. And what he found was that they pressed the button at a particular phase of the alpha wave. In other words, there could be many alpha waves between successive flashes, but whenever they pressed it at a particular phase. And it was not randomly distributed in time. It wasn't randomly distributed in time, but it was at a particular phase of the alpha.
At an upstate.
It was near the peak.
Near the peak. And so this, and who is doing this study? This is
Donald B. Lindsley
Donald B. Lindsley. Okay,
at UCLA, a long time ago.
I'd like to connect this to Lebed experiments. I wonder if like they're getting the same coincidence with alpha.
Oh, well,
that could be interesting.
Yeah. I never really understand that Lebed's experiment, but I have consciousness occurring before awareness or something like that. But anyway,
I've been working on a model that we can talk about later.
Yeah, there's a lot of evidence of that kind of thing. That, for example, that the evoked potential, the evoked visual potential from a flash of light is greatest. The magnitude of the amplitude of the evoked potential is greatest at the peak of alpha.
If it coincides with peak endogenous alpha.
Exactly. Which means that the alpha, again, that's on a purely sensory phenomenon, because it's just the measuring the flash of light in the cortex, in the visual cortex.
So if I'm understanding here, it's like you're getting this hierarchical building of, I guess I would say, like balanced order and diversity. And so you're describing like at maybe the most extreme would be like epileptiform activity, where you're getting maximal synchrony, maximal order, very large amplitude events, because everything is coordinated, but there's no diversity.
Synchronized.
Everything's synchronized, but there's no diversity within it. And
exactly.
So there's not anything to respond. It's just one thing. And you were mentioning also like a kind of almost like a window of communication, almost, but where it's like if you're too ordered, you can't listen. You only hear yourself. If you're in this like highly ordered regime of like, let's say like an epileptiform discharge, where everything's just forming this synchronous complex, but this internal signaling is so strong. If it's too strong, you can't, nothing is coming in from the outside, nothing's impacting it.
Or maybe it's coming in, but the motor system is not ready to respond. So, but maybe it's only coming in once every long interval, long intervals between whenever something is coming in, and you can't, so there's no adaptive response capability.
So there's both fewer listening windows and
exactly.
a reduced diversity of like sensitive receiver operator.
Exactly.
But then as you start to get a little bit more meaningful activity, like some sort of
diverse activity in the diverse subgroups of responsive units in the brain.
So you're getting a little bit more diversity in slow wave sleep at these like delta frequencies, but it's and so they're high amplitude, and that they're decent sized populations, like relative to like faster rhythms in terms of the numbers that are in sync. But you're also saying that it seems like they were still somewhat sparse compared to something like the seizure.
Exactly.
And so, but then on top of that, you can build another hierarchical or another like level of fractal dimension, where it's like if you're getting more meaningful activity and responsiveness, and maybe a different excitation inhibition balance, now you make room for something like alpha frequencies. And so this would be like somewhat smaller in magnitude, the waveform.
Well, it's not,
but more frequent.
It's not that it's smaller. It's that there are more of them at different phases. That was the whole point of Lindsley's thing, that it's, yeah, each, of course, each sub, now they have many more subunits, all active, all responsive, and input or capable, like you said, the windows, capable of input and capable of output. They're all these subunits have this capability at some time or another, but they're out of phase with each other, which means that you have many more opportunities in time for a response, awareness and a response. So yes, the amplitudes are smaller, but it's only because you have many more, all in at different time, at different activity cycles.
But the time, you're also saying, though, it's not just a greater spacing, but among the spacing, there's also something of a, so having more synchronous events would give you more observations, like send receive windows. But there's also potentially importantly, like a diversity within, like, let's say the alpha band of like when the peaks occur. So it's like the spacing is not like pure 10 hertz or pure, it's like it's moving within this range, but it's also within that range, it's just
No, but
variable.
I think the important thing is to compare delta to alpha, because the amplitudes in alpha are smaller, but they're much more diverse in phase. They're smaller because there are more of them, all out of phase with each other.
But the variance of the peaks is greater in alpha. It's not just that there's more, like there's more opportunities, but there's a greater variance, like if you look at the distribution of alpha over time compared to something like delta.
In the entire brain, more variance within the alpha.
Not sure if that's the case.
I don't know if that's not, it wasn't the point. It was that where in the delta you had, say, 3 windows per second. Now in the alpha, you have 10 windows per second.
So at delta 3, like that would be like in a rodent, correct? Like because
during sleep.
During sleep, but you said 3. I know like some people might like consider that to be like the beginning. Would that be like the beginning of? Would that be the beginning of theta? Like where would you distinguish between?
No, theta is 4 to 7, but I mean.
So you say 4 is theta proper.
Yeah.
And the human too.
Yeah, 4 to 7. I mean, in rats, I think it goes up to about 9. It goes for about, it's usually around 7 to 9. But if they're exploring very, very vigorously, it goes up to 9 per second. But the idea is that I don't know if it's the variability within the alpha. The alpha itself is variability. You know what I mean? Compared to delta, which
it's
is
emerging, it's faster and smaller because of the variability of the processes that give rise to it.
Exactly.
I guess I was getting a little hung up.
What?
I was getting a little hung up on the variation across upstates. I was wondering if, for instance, like let's say like you're saying, like you respond more like the flash of light if it's in a peak. But there's this was it the Nyquist sampling theorem or something like this, like in order to get like a good sampling of any rhythmic process, you have to sample at least twice. However, apparently you can cheat this if your sampling is somewhat stochastic, and you're not just sampling it like a perfect rhythmic phase for the rhythmic thing. You can get away with far fewer samples. So I was wondering, like,
yeah, there was a very clever study by Harder and White, in which what they did, and they published that in Science. That was in 1975, I think. It was really a very impressive study. What they did was they flashed the strobe at, if I remember correctly, it was 33 per second. And they measured the alpha wave in the humans. And what they did was they applied a train of strobe flashes at the rate of 33 per second, but for a half second or a quarter second. So the train length was, let's say, to make it easy, let's say the train length was half a second. Okay, at 33 per second. So that means that there was 16 strobe flashes. And what they did, they asked the people, how many flashes did you see? And if the train length was half a second, the alpha going at say 10 per second, that means that they gave 33 flashes while there were 5 alpha waves. And the people said that they saw, it was something like, what it turned out was that they only saw about 2 flashes during any alpha wave. So if they had 3 alpha waves, they said they saw 6 flashes. They could count, they couldn't count 10. But so they never, in other words, if they never said that they saw, there were 16 flashes in a half second, but they never said that they saw 16 flashes.
We're saying only like 2.
They saw five or 6.
They said 5 or 6 during a half second.
During a half second.
So it had so basically they're only seeing it when like they're counting it basically the upstates of alpha.
Exactly.
And it has to actually correspond to it and be within that window. So they might drop a few if it's not arriving.
They dropped them when it wasn't near the peak of the alpha. So, if they did it for a quarter of a second, they only saw 3 or 4 flashes, even though, there were 8 flashes shown, they only saw a few, but they said that, they're not rhythmical. They just saw these flashes.
So that's the direction I was trying to move into, because I was wondering like this variability and like the sampling from the environment.
That's
and
right. They're sampling.
Sleep, you don't need to be sensitive. You don't want to be sensitive. You're like, it's endogenous.
Well, except
unless you wake up
if you hear a baby crying.
Yeah, like unless it's an emergency, yeah. But otherwise, like want
not
like
an emergency. Just certain things come through. My friend Norm Adler did a study in rats where he played ultrasounds of the. He recorded the sound of pups squeaking in ultrasound.
That'll be a high bandwidth channel.
What?
That'll be a high bandwidth channel. You want to keep that one open.
Yeah. And then he played that to the maternal rats when they were sleeping. And they woke right up.
Let that through. Make sure that can get through the blockade.
Other sounds, even of higher intensity, didn't wake them up.
I noticed you skipped though from delta to alpha over theta. Is there a is there a different principle at play that made you want?
No, as a matter of fact, I did one study that I really I feel very proud about, based on this with theta. That we recorded I recorded the theta in rats, trained them to press a lever for food, and so it was ad lib. They could press the lever whenever they wanted, but I was recording the theta continuously, and what I found was that, and we had the switch from the pedal synchronized on the same timeline as the theta, and it turned out, and my null hypothesis was that they're going to press the lever at random phases of the theta. That was the null hypothesis. In other words, they would press it independent of what the theta rhythm is. They would press it at any time. It turned out that there was highly significant that they press the lever at the peak of the theta, and they release the lever at the valley of the theta. We published that. It was really striking that the initiation occurred at a different phase. It occurred not randomly in time, but definitely was a highly significant.
Initiation and maintenance, or would you say like the
It
maintenance
wasn't
comes for
maintenance,
free?
just what? It
Would
wasn't
you say the
maintenance.
maintenance comes for free? So it's like you're pushing down the lever.
Didn't come for free.
Didn't come for free.
It didn't come for free unless that's the reaction time or something, it just happened to be that way. Maybe.
I guess what I'm wondering is it like, so as you're going into upstate, this is initiating like the push, but
Yeah.
then, so I guess what I'm wondering is it like, to the degree you're getting this theta coherence, this theta orchestration, you're able to maintain it, and then as that attracting state goes away, you just take your hand up, or is it kind of like at the peak state, some sort of ballistic process takes place?
Yeah, it could be either one. It could be, that's what I mean. It could be ballistic in the sense that it's an invariable amount of time until they release the lever. It's just sort of a ballistic pattern. But that's what happened. I mean, we got a significance of when they pressed, a significant phase relationship when they pressed, and a significant phase relationship when they released the lever. Even though there were many seconds, there could be many seconds in between successive lever presses. And of course, many intervening theta waves. So the same exact principle holds for the theta, at least in the rats. And of course, the earlier study that I did with the correlational study that this was based on was that the sniffing behavior, the vibrissa twitch and the inhalation are synchronized with the theta, with individual theta waves. They all go at 7 per second. And when they really get into, when the rats really get into intense exploratory behavior, and what I mean by intense exploratory behavior is they're standing at either mainly when they were standing at the edge of the table, looking over the edge, and not, just sniffing the air, looking over, it looked like they're trying to decide what to do. That's when we got the highest amplitude theta, the highest amplitude, most regular theta. Also,
under these conditions of some uncertainty. And so it seems like the theta is indicating either
uncertainty, but not only uncertainty, but looking like they're trying to make a decision. And also,
uncertainty that you want to resolve for the sake of action.
And the other time we got the maximum amplitude and regularity of the theta was when they were standing up on their hind legs against the wall, sniffing the top of the cage.
But they might also be doing okay, so if I'm understanding, the theta, the evidence is suggesting is this coordination of action, including active sampling of the environment to forage for information. So if you're like sniffing, that's kind of action. If you're using your vibrissae with like your little blind person canes, that's a kind of action. And also, if you're sniffing like up top, so you're trying to get this information. It's of higher value.
Olfactory information and tactile information jointly, because the sniff and the vibrissa sweep going forward generates tactile input from the floor, from the surface. So it's generating tactile input simultaneously with the olfactory input.
That has to be like the strongest source of entrainment an animal could get, almost like you're getting like all of the like from the whisker barrel areas, like all the vibrissa at the same phase coherently with this, or
at some link, at some linked phase relationship with the olfactory input,
and at the same time cross-modal, but not just any modality, but like olfactory with like its hard line, like to amygdala, hippocampus, like it's coming in very centrally.
It's
all the same.
Correlational. Make the difference between a piece of food and a rock.
it just seems like the mind has to be built on top of this. Like this
That's
has to
right.
be like the
That's
dominant
exactly
attractor. That you,
right. Right.
this is the starting place. This has to be the
Right.
starting
Not
place.
only not only that, but when they're really getting into it, the heartbeat is going at exactly the same rate. They're all 7 per second.
Yet, so yes, it's like we're talking
And
about like,
the heart, this is wonderful study by Satchel in Dogfish Shark that she showed that the shark takes a gulp of water into its mouth at the same rate, but at a slightly different phase that the heart beats, and that the tail swings, all 1-to-1. So 1-to-1 ratio, that the gulp of water coming over the gills, she calculated that the rate of flow over the gills is precisely synchronized with the rate of blood flow through the gills. So it's getting optimal oxygenation by taking the gulp of water that's passing the oxygenated water passing over the gills, and the heart and the blood flowing through the gills. It's getting optimal oxygen input.
so you have this constraint for trying to establish as much optimality as you can in movement and swimming, because if you're not a good swimmer, you're not going to last long. But now this is a constraint on your
And
sampling from the environment. This is a
and
constraint
and
on
the swimming
something
movement,
else.
the whole bodily swipe from side to side is at the same rate as the gulp and the heart rate. So that's like a little robot of energy optimization for movement, because it's getting the oxygen to the blood, the oxygen into the blood to the muscles of the body, the whole body, to swim.
So,
Like a little machine, like a little damn robot.
so this is a wild one. To what degree would you say that this requirement for like harmonious or like harmonic functioning mechanically, to what degree did that require or scaffold a rhythmic neural syntax? To what degree was the meaning of
Scaffolded it. I love that word. That's a great word. It's the primordial. First you have to have the optimal energy machine, like in the shark, where you get the oxygen, the blood flowing to the muscles, the oxygen delivered to the muscles, so you get the optimal movement. In the rat, you have the air coming in synchronized with the heart. That's getting oxygen into the brain, but it's also doing the processing, the cognitive processing of the environment. So, building
so the
on
maximal flux of blood into the brain, you coordinate this with the sniff also, so that the oxygen
with the
is there.
sniff and the touch, with not only the sniff is bringing olfactory information, but it's also bringing air. It's
So you
bringing
want the
oxygen.
yes, if you're going to be forcing blood through the brain, you want this to coincide with good oxygenation. Otherwise,
Exactly.
you're it's going
And
to be
if you want
massive.
optimal processing, you want the neurons to be optimally functioning, so they get their optimal oxygen from the sniff, and they're getting the olfactory and the tactile input to make the decision. So it got the whole brain doing just the right thing. The whole body is doing is optimizing decisions. They have a relatively small brain. You can't afford to, well, I mean, it doesn't always work. I'm saying that is when they really have to make a decision, like are they going to jump off the table, or are they going to jump up or out of the cage? That when they're standing up or they're leaning over, they're at a decision point. The little animals are making a decision. If they're running along the table, there's no theta. Talk about ballistic. That's just ballistic. But when they get to the edge of the table and they look over the edge, that's when the theta starts. so it's so they have, so of course they don't always have the theta when they make, I mean, they can the synchrony is not always optimal. It's not always this beautiful thing. It's only at the extremes. That's when you see that beautiful synchrony. Otherwise, it's relative. It's like relative oscillators, relative phase linking. Oh, it's 11:55. I'm going to get a phone call in 5 minutes.
Okay, so we have to continue this next week.
Yeah. Okay.
So we're gonna be okay. So I like where we're heading.
I mean, this is something I've been thinking about forever. It's ever since I made the I was in Jim Old's lab, trying to see if I can correlate neural activity with behavior. That's what he asked me to come out and look at, to, listen to the neurons firing and see if I can see what the animals are doing, if there's any correlation. And I noticed that there's this terrible artifact when they're sniffing, and it's interfering, the facial movements, the sniffing movements are interfering with the hippocampal theta.
Might have been the golden thread, that artifact.
That's right. That's right. So I realized that, wait a minute, this is the limbic system, the olfactory brain, the theta rhythm is synchronized with the sniffing. I got so excited that night when I made that observation. That's a whole story. I went to the cafeteria at midnight with my notebook at University of Michigan, where I was working with them that summer. And I was writing and writing notes, and the night watchman came over to me and he said, "That you see that sign over there, buddy?" I said, "What sign?" He said, "Go take a look at that sign." And the sign said, "There's no reading or writing in the school cafeteria after midnight." He said, "It's after midnight and you're reading or writing in the school cafeteria." I said, "There's nobody here. It's this, this giant cafeteria, the capacity of 500 people. There's nobody here. I'm not bothering anybody. I'm just sitting here writing my notes."
For your own good.
You better stop it, buddy. And he said, "Or I'm going to call the cops."
But it would have been for the rest of ours good. Yeah.
What?
Maybe it was supposed to be for your own good, maybe. But I'm glad you did it.
Yeah. I found out why they said that. But anyway, so he called the cops, and they arrested me.
That is dedication to science.
The Ann Arbor police arrested me for writing in my lab notebook at night, after midnight. The next morning, Olds told me I was crazy. Don't mess with the Ann Arbor police. And I found out that the reason from the dean, I found out the reason why they have that sign is that if they let students read or write in the school cafeteria after midnight, then they'd all leave the library and come over to the cafeteria. That's why I have the sign. That made a lot of sense.
Okay, so it was less looking out for the students than I thought. Okay.
Right. Anyway, I'm gonna get this call at noon.
Okay.
So,
thank you. Let's keep going, guys. Same time
same
next
time,
week.
same station. Okay.
Yeah. Take care.
Take care, Ann. Bye.